Hi, everybody. Welcome to today's installment of the Spark Seminar Series, Bridging the Body and the Brain. I am thrilled to invite you to join in this conversation today with our members from the Spark Data and Resource Center, Dr. Esra Neufeld, Dr. Antonino Cassarà, and Dr. Javier Ordóñez. And today we are going to be diving into in silico safety assessment of neural interfaces. So this is going to be just a spectacular presentation. I cannot wait to dive in. So let's get to it. Before we get into giving the floor to the IDIS Foundation team, I just want to share some information about some exciting activities that the program, Spark Data and Resource Center, are doing to build the Spark portal into a community resource for all of those interested in neural organ control and somatic control of the body. And so to that end, we are excited to announce a partnership with Autonomic Neuroscience, Basic and Clinical, where together we are hoping to integrate data deposition into the publication process so that we're advancing both of our missions to make research more transparent and impactful. We're thrilled about this opportunity to make data and the data interpretation as reported in the publications just that much more visible and tightly aware. So if you're planning on publishing in Autonomic Neuroscience, which I hope you are considering, that you will also consider depositing your data and models on the Spark portal to create that linkage between the interpretation and the data itself. As I said, today is our latest installment, and just a heads up, tomorrow we're going to host Dr. Von Macefield at 11 a.m. Eastern, so same time, same place, just 24 hours later. We'll be talking to Dr. Von Macefield about his intraneural microelectrode recordings from the human vagus nerve. It's going to be really fascinating. I'm super excited to have an opportunity to chat with him. Not only that, but the following week, so a week from today, but at 3 p.m. Eastern, so Dr. Marianne Martone and I will be diving into our research on how we're creating a knowledge base of peripheral nervous system connectivity. So that is a feature of the Spark portal that brings data and knowledge together and surfaces it for exploration. And we're going to dive into how we create it, how we're using AI to populate it and surface information, and get into the nitty-gritty of what we can actually do with this information as a source for preliminary data for your grant applications, just to explore what you know, and even for experiment planning. And so we look forward to having that conversation a week from today. So the Spark portal is an open neuroscience and systems physiology platform. So we're aiming to bring together the physiology, the anatomy, and devices together with various types of modeling all together into a single platform so that each scientist each scientific discipline can be represented and have an opportunity for enhanced collaboration. We are a NIH-approved HEAL compliant repository, so you'll find nociceptive data sets represented on the portal, with more coming every day. And additionally, one great feature of the Spark portal is that we have been accepted into the AWS Open Data Sharing Platform sponsorship program. And so what that allows is free egress and storage of all data that is published to the Spark portal. So if you are considering where to put your data, once it's published on Spark, it is freely accessible for you So you're basically future-proofing yourself. You're thanking your future self will thank you for making your data freely available and understandable for the long haul once it's on the Spark portal. So what do we have? We have over 65,000 protocol views. Each data set that is contributed to the Spark portal includes a detailed experimental protocol if it's an experimental data set. Obviously, if it's a computational model or an anatomical model, they will not have a protocol. Data sets themselves have been downloaded over 10,000 times. We have over 2,000 active OSPARC users. And so Esra and company will be diving into all of the things that these people are able to explore and use in just a few minutes. And these data set citations are growing every session. And so what can you do with Spark? You can browse, view, and get data and models. You can view the context of these data sets and models within an anatomical context, which is set up for various species, male and female human, and you can see which data sets are all together within a particular anatomical location. We're diving into how computational pipelines are created today. And then lastly, we encourage you to contribute as well. Help build the community and help create a mechanism for enhanced collaboration with the data and models that you produce. It's a living, dynamic system. OSPARC is free. Please request your free account by sending an email to support@osparc.io. And this will allow you to port data from the Spark portal into OSPARC for creating your own analysis pipelines. You can bring in your own data, and the models that you create can be generated or packaged into services that allow others to use what you've created in a safely packaged manner. There is a new publication that is available right now on bioRxiv that describes how OSPARC was generated and created and the reasons why the materials were created as such. And just one last plug for why you should consider contributing to Spark. We accept data sets individually. So, you know, an experiment that you're doing right now, you can contribute to the Spark portal. It will help you build your body of work. You can deposit your data and reserve your DOI so that it goes live at the same time that your publication goes live. And additionally, it can include all of the components of your experimental application because the Spark portal is a domain-specific repository. So we are interested in PNS and systems physiology domain, but we're agnostic when it comes to the methods of data acquisition. So code and simulations can sit side by side, very large image data or physiology or RNA-seq data. So it gives a great way for all your experimental information to be packaged together. And when it comes time for you to submit your next proposal request, you can actually write us in as indicating that you are planning on submitting your data to the Spark portal. We provide grant language assistance, and depending on what type of relationship you would like to have with the Spark Data and Resource Center, we're perfectly excited to consider consortia support. We have various custom services, which will be highlighted today with Esra's presentation. So as I said, there's so much data available on the Spark portal. There's over 11,000 samples across 4,000 subjects, 26 methods, 54 tissues and 7 species, all from 600, more than 600, 615 as of today, unique contributors to the portal. So your data is welcome here. Your models are welcome, your devices are welcome. Come join us. As I said, I just said this, we've got 250 research data sets, 47 anatomical models, and 46 computational models, and there are 100% available for your reuse. So you're not just putting your data someplace on a shelf, you can actually recombine your data with other data to generate new insights. Your data is accessed. The data sets that you contribute are used, and often they're used by the people who contribute them. So it's great to know that your data is easily accessible and findable right here on the Spark portal. You can see that regardless of when data sets are contributed along the x-axis, that data sets have quite a bit of activity regardless of what type of data set it is, computational model, anatomical model, or research data sets. And with that, I'm going to hand it over to Esra.
Yes, so now it should be right on. Yeah, we'll talk about modeling as in silico regulatory evidence, and I will be providing some background on that, on the requirements, on the possibilities that nowadays in silico modeling offers to support regulatory submissions. Then we will have a brief introduction of the OSPARC platform, which is the basis for the modeling that we will show today, before Antonino will show you the neural interface modeling pipeline that we have created. Javier will provide you with the necessary background and also a live demo of the meta modeling framework that we will be using for systematic uncertainty quantification. And then before wrapping the whole thing up, I'll be talking about illustrative application of the methodologies in the context of an in silico safety assessment study we did for a generic neural interface, a certain cuff electrode. So in silico as regulatory evidence, computational modeling has an increasing role across all the stages of medical device development and use. Computational modeling offers rich insights and dense information that is not necessarily experimentally accessible to analyze a certain application. You can use modeling to optimize the design of a device, its safety, its performance, to elucidate the underlying interaction mechanisms or identify potential problems with the device. But in silico prototyping and testing is then also a very powerful means of reducing the time it takes to get a device to market, to perform trials. In silico trials can complement or even to some degree replace animal and human trials, something which is particularly interesting with regard to the latest NIH directives that I assume many of you have heard of. And then computational modeling also serves to support precision medicine by allowing personalized treatment planning and treatment optimization. Now, when you use computational modeling for regulatory submissions, the typical applications of regulatory submissions are the demonstration of safety and of efficacy. And the FDA, as well as other regulators worldwide, are now actively encouraging the use of in silico evidence as part of such safety and efficacy evidence and demonstration. For that, there have been big efforts by the FDA, but also by the ASME VNV40 committee, for example, and other committees to standardize the reporting and the efforts to be done on the verification and validation side of things. And don't worry, I'll explain the difference of those terms in a moment. Now, computational modeling, there can be different degrees of decision-making that depend on it. It can go from identifying the worst-case configuration that is subsequently tested experimentally, in which case relatively little depends on the modeling, but it can go all the way to replacing the trials, and modeling can even become part of the algorithms of the controller of a device, in which case much more depends on the modeling. And the degree to which verification and validation must be performed is commensurate with both the risk and the credibility associated with the modeling. So when I use the term verification, what I mean is ensuring that the implementation that you have of a model is in fact implementing the model that you want to implement, the algorithm that you want to implement correctly. The validation, however, is about ensuring that what you want to implement actually captures reality, and more precisely, that it captures the relevant behavior of your quantity of interest within a given context of use. Those are all keywords that we will be coming back to. And so if there is a big risk associated with the decision-making supported by your modeling, and if a lot of the decision is taken based on the credibility of your modeling, these two factors, risks and credibility, define the degree of efforts that must be performed to ascertain that the modeling is indeed credible. And there are various aspects, from the convergence over the appropriateness of your model, over the consideration for variability and uncertainty, where you can do different degrees of efforts to prove that. This is a table out of the ASME standards draft. And depending again on risk and credibility, you need to do more or less such efforts to establish the credibility. So here the keywords again, you're trying to predict certain quantities of interest that are relevant with regard to safety and efficacy. You need to show that you capture that within the expected uncertainty. That's the first mention of uncertainty here, and within a certain context of use. So any model comes with assumptions and simplifications that are only appropriate in certain contexts of use, and the validation therefore is also limited to a certain context of use. And once you leave that context of use, your validation effort needs to be repeated to demonstrate that your model is still appropriate. And in that context, sensitivity and uncertainty analysis are key. So sensitivity analysis serves, first of all, to identify critical parameters, parameters of your model where you need to have a high degree of control. And if you don't know those parameters enough, you need to have a bigger effort in either controlling or in identifying those parameters. Sensitivity is also important to know how powerful your validation is. For example, if you have a validation experiment that only insensitively depends on your modeling, then that's not a good test, because you might be way off with your modeling and your benchmark will not pick it up. So you need to do an uncertainty and sensitivity analysis to understand the strength of your validation. Also, to decide whether the validation is in fact in agreement with the model, you need to check that that agreement is within the expected uncertainty. So if you have a small expected uncertainty, even a medium deviation will highlight a problem with your model. However, if you have a large uncertainty, that means that you have a model that has maybe little strength in what it says, but it's easier to show that it is validated within the expected uncertainty. So we have a need to understand the safety margins and the associated uncertainty of our models, and no validation effort is complete without uncertainty assessment. Hence, the key focus will put on uncertainty quantification in today's talk. Frequently, what you want to do is you have a certain model, and that model is combined with input parameters that each of those has associated uncertainties. And you want to propagate that uncertainty through the model. So what that means in mathematical terms is there is a certain probability distribution for the underlying parameters, and there is an associated prediction from your model, and you want to know the variability, the standard deviation of your model, considering the underlying uncertainties of those parameters. Now, if your model can be linearized, so if the model is dominated by linear dependencies on the underlying parameters, and if the probability densities of the different parameters is independent, it's easy to show mathematically that you can decouple the analysis of the uncertainty of the different factors. You can determine the uncertainty and you can combine those using a root sum square approach. That's a very common thing to do. However, as soon as you have dependencies, so here we have a very simple model where we are looking at the product of conductivity and permittivity, but both of them depend on water content. If you use this linearized approach, you would come to the formula that I'm showing here, and you would predict a certain uncertainty that is wrong, because you are neglecting the fact that you have correlations, that two of your parameters depend on the same underlying parameter that is uncertain. And instead, accounting for those interactions and those correlations leads you to a correct uncertainty that is 40% higher than what you would have thought with a classic analysis that decouples those. So what we need to do instead is, when we have this kind of nonlinearities or correlations, we rely on methods such as the Monte Carlo method, where you sample your parameter space according to the probability, and you assess how your function, how your predictions depend on those uncertainties. That is slow to converge, and you will often, when you have high-dimensional parameter space, you will require tens of thousands of model evaluation to converge. And you don't even have a guarantee that you converge. Instead, if there is an issue with ergodicity, it might be that you always explore the same subpart of the parameter space, and that you always stay in some subregion, and you never cover the whole probability space, and you get wrong predictions. So we need a way to explore the whole parameter space, even though that parameter space can be very large, without requiring tens of thousands or millions of costly computational model evaluation. And that's going to be the topic of the part that Javier will show you, the use of surrogate modeling and of intelligent design of experiment methods to render such analysis affordable. And before I hand over, just briefly about the platform. So we are talking about the OSPARC platform that was developed in the context of Spark to host all kinds of computational models, to power them, to allow sharing them, evaluating them, publishing them, and documenting computational models. The platform is a cloud-based platform that has been developed to support the modeling work of the over 100 Spark teams, and to ensure that those models can be sustainably preserved, that they can be integrated into overarching models, and that they become reproducible. They should be openly accessible. They should adhere to FAIR principle. FAIR stands for findable, accessible, interoperable, and reusable. And we want them to be scalably executed, shareable, and collaborative. And what's key is we didn't want to develop a platform that disrupts established routines, because we know that people will not adopt methodologies that are contrary to the established methodologies in the lab. Instead, the goal of this platform is really to allow you to continue using your tools, your modeling approaches, but to do so on a platform that allows you to then have reproducibility, interoperability, and fairness built in. And the challenge of fairness in computational modeling and of reproducibility is that frequently you have runtime environments and operating system dependencies and changes over time. And when you come back 5 years later and your PhD student is not here anymore, you suddenly might not be able to reproduce the work that you have done. You also might not be able to remember exactly what was done. And it's also hard to work with others that might have complementary approaches, but very different tool sets. So what we are doing with OSPARC is that we are packaging the models along with the environments in which they were created and executed to allow a traceable, reproducible, and documented form of running these models. You can build pipelines out of different models. There are over 100 different services around nowadays, ranging from simulation tools to data analysis and visualization tools, AI tools, and much more. And when you have created such a pipeline out of different modules, you can package it. So this is an example of a modeling pipeline built to allow planning of spinal cord stimulation to restore locomotion to paraplegics. And once this complex pipeline was built using collaboration from 5 different teams all over the world, this was packaged into a step-by-step application suitable for clinical deployment. And this kind of application in guided mode is one of the other benefits offered by working on OSPARC. And so OSPARC has built-in support for verification, validation, and uncertainty quantification with powerful tools for uncertainty quantification that Javier will show you, as well as support for information standards such as the 10 simple rules, which are ways of documenting your verification and validation tasks, of ensuring that the context of use and the limitations are clearly documented. And we have support for these 10 simple rules also in OSPARC and in the minimal information standard that is part of the SPARC data standards 2.0. And with that, I'll hand over to Antonino to show you the neural interface modeling pipeline.
Okay, so what I will show you now is how we implemented a modeling pipeline that allows to investigate the safety and eventually also the performance of neural interfaces. The idea is very simple because we are in a world where we are developing a lot of technology that aim at interacting with the nervous system for sensing application, but also for stimulation and closed loop control, and the possibility to optimize and verify the safety of these interfaces applied to different systems. For example, different nerves or with different interface for a specific nerve is very, very important. And the nervous system is very heterogeneous in terms of properties of tissues, but also in electrophysiology. So we would like to test as better as possible this implant sensor or neural interface on different nerves. For example, we would like to test how a different new approach of stimulating a nerve, for example, via some catheter electrode or with soft neural interfaces. You would like to test all these new applications. Therefore, we created this pipeline. Sorry, maybe I can remove this. Okay. So we developed this pipeline in OSPARC that allowed to develop, first of all, histological images, models of complex nerve based on histology data provided by SPARC team or also provided by different users. And this pipeline allowed to create complex D or 2.5D models of nerve that can be used, can be neurofunctionalized first to model the neuroelectric response of complex nerve to electric stimulation, and therefore you can test how a specific implant or a specific neural interface behave, and especially if it's considered safe for the specific application. So in the specific application that we develop, we use data, so nerve cross-section images, as well as quantification of axon type, axon diameters to create 3D complex model of porcine vagus nerves. These data were provided by the FINES team, and then we develop, based on the pipeline that I will show you, we executed the electromagnetic, thermal, and electrophysiology simulations to predict electromagnetic exposure, first of all, and then couple this electromagnetic field distribution, energy density, with thermal simulation to predict heating of the nerve and the tissue surrounding it. And then we coupled with the electrophysiology model of individual axons to predict threshold for stimulation for a specific part shape. All this information, all the simulations have a specific goal. The goal is to quantify quantities that are relevant for safety and also eventually for performance. For example, average current density, field strength that are relevant for international standard, international exposure standards such as the ICNIRP guidelines and the EEE, or the quantity like charge accumulation, the quantification of charge density, and then therefore the Shannon criteria, tissue heating, and as said, a recruitment curve that quantify threshold current for specific pulse stimulation. So we start from, you see in the image from nerve section images, and in the specific application, we create 2.5 or 3D nerve model based on the pipeline, based on the images that is shown on the bottom. So we extrude several, we create a surface mesh on a 2D cross-section image. We create electrode in the specific cross-section image, in the label image, and then we create a surface mesh that we extrude in different steps to create a simplified model of the nerve with a specific device. And what you see here in the image below, those are different cross-section of electric field, current density, energy density, and then also temperature increase. And so we use technologies that are available in C4Life, and that is available with sparc, using also other functionalities, for example, libraries for computer vision to extract, for example, geometrical information from the labeled image. As I said, we calculate also the Shannon criteria, but we will quantify some of this conclusion later. What I will show you now in this moment, it will be a live demo. Expected. Okay, so what I show you now is one of these complex 3D nerve model that we generate. How we generate this? This is the pipeline that I was introducing you before, and I will briefly describe. So first of all, in the, as you can see, as Esra also described, is a pipeline built with different services. Every service performs a specific function. So we provide a data set. So from the quantitative database, it means the cross-section of the nerve and also the axon segmentation information. Then we have services that do the model generation by doing some geometrical activities on the nerve, for example, contour extraction, adding some padding, skin layer, adding a skin layer to create electrodes. We have services for creating a 2D mesh on the generated nerve cross-section images, extruding 3D, then the created surface mesh to create complex unstructured mesh, co-registration of the axon data. And as we finish this model generation, automatic model generation, we have a complex 3D model that is the one that I'm showing you. So here you see, this is the nerve, this is a surface mesh generated on the nerve cross-section image, as well as the electrodes. If we move a little bit around the nerve, you can see that when we use the pipeline, in this case, we applied bipolar cuff electrodes, okay, in the middle of the nerve, and then we will apply to this bipolar cuff electrode some applied voltage and a specific pulse shape. So when we finish the model generation, then we have the solver, so the physics. So we calculate, first of all, the electromagnetic field exposure, including also quantities of interest, for example, the current flowing through the nerve, outside of the nerve. We calculate energy deposition, and then these quantities are used in the thermal simulation to calculate the temperature increase, and in the electrophysiology simulation to calculate the amount of individual fibers that get stimulated for a specific pulse shape. In this pipeline, you can also see that there is a function that is also this report generation. This one is a Python notebook that, for example, you can customized to extract an automatic report generation. And just to conclude my part, I want to show that you can also, if I can, that you can also modify this pipeline in such a way that you can use the meta modeling activity, the meta modeling pipeline to perform all this meta modeling activity that Esra was mentioning before, and that will be the topic of my colleague Javier. And with that, I will stop.
Thank you, Antonino. So Esra will go over the results of this pipeline later, but first I would like to introduce the meta-modeling framework. What do we mean by meta-modeling? At the end of the day, a model is a simplified version of reality, but it can still be very expensive to evaluate. When we include high degrees of realism and accuracy, as in the pipeline that Antonino demonstrated, every evaluation takes several hours. In numerous applications, such as sensitivity analysis or uncertainty quantification or optimization, we require to repeatedly evaluate these models. And when the number of parameters increases, the number of required evaluations very quickly increases very quickly. And this becomes prohibitive in terms of time and resources very fast. Meta-modeling is addressing this issue by constructing an approximate model, SUM for short, of the full model based on a very limited number of samples. How do we choose those samples? Well, we use strategies from design of experiments, for example, light and hypercube sampling, and then we can further refine those sampling strategies using data-driven strategies. For example, to reduce the uncertainty of the model or explore critical regions such as optimal performance regions or potentially safety, like failure regions. So once we have a parameterized study in sparc, as Antonino demonstrated, we can evaluate it in parallel using the inherent cloud scalability of sparc. And then we can use the suite of algorithms built in the Dakota library developed by the Sandia National Laboratories to perform a variety of meta-modeling approaches, such as building a surrogate model for response surface inspection, performing optimization, performing sensitivity analysis and uncertainty quantification, etc. Now I will demonstrate this approach with a tutorial we have built for the Sparc platform. It concerns the design of an implantable neural interface on the sural nerve. So first we will, I don't know if you can see my mouse, but we will inspect the effect of the design parameters to choose an optimal configuration. And then based on that optimal configuration, we will assess the impact of the tissue, of the human tissue uncertainty to, as Esra mentioned earlier, validate that this is truly a safe and efficient device under uncertain conditions. And for this effect, we have developed a suite of tools that are available on sparc, which allow to explore these results interactively and guide the user through the different steps. So now I will switch to the live demo. Sorry about that. Okay, now you should be seeing things again. So here, these tools are available under the, if the screen allows me. Yeah. any user can come here under HyperTools and access the preview of these services and follow the tutorial that was shown in the previous screen. And for those users that are interested, we can enable the full version to run these tools on their own pipelines. Once we have started this, we can, for example, choose the function that was created based on this pipeline, set some parameter ranges. For example, in this case, the design parameters were the width of the electrode in terms of how much it covers of the nerve, how much space do we leave between the electrodes, how long are the electrodes themselves in millimeters, and how much padding of the insulation silicone we leave around it. Then, in order to measure whether we can use the surrogate model, we must first validate that it is accurately describing the observations. This you can see here, that the distribution of the predictions is very closely resembling the distribution of the observation, and therefore our takeaways from the surrogate model will be accurate. Because we have a 4-dimensional input parameter space, we cannot visualize the full dependencies. Instead, users can inspect cross sections to see the dependency of single variables, or in 2D or 3D, that is inspecting the quantity of interest, in this case, the current necessary to activate 50% of the neural fibers, based on the different design parameters. So for example, here, we can quickly observe that the width of the electrode has a much bigger impact in reducing the necessary current than the spacing between the electrodes. This we can also do in terms of 3D isosurfaces. And we can see that having a bigger interelectrode spacing, having a big coverage in terms of width, and having a low coverage in terms of length is what gives you optimal effectivity. However, if we look at the safety criterion at this current level, which is the Shannon criterion, we can quickly get a prediction for this thanks to the surrogate model. And we see that while the dependence on interelectrode spacing and width is the same, we need length is positively correlated with safety. Therefore, we must choose something intermediate. So once we have chosen based on all this data and optimal configuration, we can proceed to do uncertainty quantification around that optimal design. For this, we implemented the uncertainty quantification service, and based on the ETIS database of tissue properties, we select the most likely tissue conductivity for the blood, the connective tissue, and the different parts of the nerve, and establish a 20% standard deviation around each of them. This enables to obtain an histogram of the uncertainty of the value. The mean will be close to that predicted beforehand, but also we can see how much the fact that we have uncertainty in the tissue parameters is affecting our predictions. So for example, for current, we can see that we have around 10%, 12% standard deviation, even if the inputs are 20% uncertain. And we can validate that indeed the model is fairly accurate. With this, I will give the floor back to Esra, that he will share the insights on how we used this to assert the safety of a full device.
Thank you very much. For the sake of time, we'll have to skip over some parts. some key observations that we do in these results. First of all, we see the obvious effects of dielectric contrast in different materials and why some parts have higher currents, while some parts of the nerve have rather higher fields. We also see that while the electric field is highest next to the electrode, in this case, because of the energy deposition in the fascicle that is high, the heating is in fact more centralized. That's also due partly to the cooling through the surrounding tissue. We get insights into how about 30% of the current leaves the device and flows around the device. Something which can be influenced, for example, by changing the length of the cuff, and that depends also on the presence nearby of important blood vessels, for example. And we get an idea about the variability across different subjects. We find that there are important correlations that, for example, dosimetric exposure quantities can be inversely correlated with the amounts of current needed to activate certain fibers, but that high degree of activation depends much more on the statistics of fiber distributions in the nerves. We can't go into the detail of that today. And we use the data here in this application from Spark. So we use data about cross-sections of porcine pig nerves, including variability and statistics about the fiber distribution in order to perform a full assessment of an interface and its uncertainty. The different factors of the uncertainty that were studied are inter-subject variability. For that, we use at a different histological cross-section to create a multitude of nerve models and to quantify the impact of that important variability on the different predicted safety quantities. We also asked the question how much of an influence the environment has. Environment in this case is the detailed anatomical environment. So the nerve is implanted within a detailed anatomical head model to have realistic dielectric environment, but we also account for the formation of fibrotic tissue about scar encapsulation around the interface. We find that the 3D geometry and the environment have quite an important impact, but that, for example, the scar tissue formation, which you see in the top table, is usually changing things only below 1%, except for the quantification of unwanted exposure around the implant, where the encapsulation has an important impact. However, the anatomical environment itself can have up to a 3-fold impact on dosimetric exposure and other safety relevant quantities. So it's important to account also for the anatomical environment. Another aspect of the uncertainty concerns tissue properties, where we parameterize 11 different tissue properties based on statistics from literature study about different tissue properties. And while this is an 11-dimensional space that could easily require tens of thousands of simulations, we leverage here the methodologies presented by Javier. So we built a surrogate model. It turns out that less than 100 simulations are needed to produce a very accurate surrogate model. I will quantify that in a moment. We get response surfaces. We propagate the uncertainty of the underlying parameters through that response surfaces using the techniques presented earlier. And we can identify again which parameters have important impact. For example, the epineurium on the amount of currents that flows, or the insulation of the fascicles on their ability of being recruited, and other parameters. So this surrogate model gives a very detailed impact assessment of the very nonlinear responses to different material properties on heating, on recruitment, on exposure, and on tissue damage metrics. It turns out, for example, that the electromagnetic properties can have an impact of about 4 dB on quantities of interest, between 1 and 4 dB, but the thermal properties have a much smaller, only half a dB impact on the underlying uncertainty. We also can look at numerics, at the convergence of the sampling, at the convergence with the number of fibers simulated. All of this turns out to be very well under control and to have a much smaller impact than some of the uncertainty factors that we have discussed. When all of that is put together in a combined uncertainty budget, we see that the dominant factors are the electromagnetic properties, shown here in dark green, the intersubject variability, which is shown here in the blue parts of the uncertainty, as well as the anatomical embedding that can very strongly influence some of the other factors. And altogether, we can end up with uncertainties in the range of a factor, let's say 1.8 for some of the most uncertain predictions, whereas other aspects such as shunted current have hardly any uncertainty associated with it. So this kind of methodology allows you to understand which factors are important, how they influence it, and that then leads us in a final step to the safety prediction. So in terms of heating, what we find is that single pulses at sufficient spacing would have heating that stays well below 0.1 degree, which is the lowest threshold the FDA is known to use for nerve tissue safety assessment. But when you have continuous stimulation and when you aim at a high level of recruitment, heating can exceed 2 degrees centigrade, which is definitely a safety concern. The Shannon criterion, which is a very questionable safety criterion, but still a commonly used one, has a similar picture. If we aim for a high degree of fiber recruitment, the Shannon criterion suggests that we're not on the safe side, considering the uncertainty margin that we need to include. When we look instead at exposure of surrounding tissues, the picture is a bit more mixed. So the exposure does exceed what would be considered safe based on standards such as low frequency exposure safety, but it is much lower than the nerve exposure. And in view of the therapeutic benefit of the nerve exposure, I think that it can be well argued that this can be tolerated, seeing that provided that there are no other important nerves and nerve fibers in the vicinity of the target. In conclusion, what I want to highlight is that we have, thanks to surrogate modeling techniques, an acceleration by 8 orders of magnitude. So that's in the order of 100 million-fold acceleration. 50 to 100 LH samples sufficient to cover an 11-dimensional input space, which is ridiculous if you think how many points that is per dimension. That's less than 1.5 points per dimension. And if we would have neglected the nonlinearities, we would have had a far off uncertainty estimation. So the use of a complex nonlinear method was crucial in doing these kind of predictions. And also the ability of OSPARC to leverage cloud-based resources means that such an analysis can be performed overnight using parallel computing resources. In future work, we will leverage the powerful and rich data that Spark researchers from REVA are acquiring on human vagus nerve to repeat this analysis also in humans. We also have the possibility of including different neural interfaces on this pipeline or other physics than electromagnetic stimulation. So here you see some of the modeling methodologies built in for acoustic neuromodulation, and the pipeline can also be used then to optimize selectivity, power efficiency, and safety of such devices. And to close, we have a comprehensive pipeline for in silico assessment that uses sophisticated surrogate modeling technique for uncertainty quantification, which ingests the rich data produced by Spark and yields quantitative insights into the biophysics of current flow, the important parameters for stimulation, and that are the basis for thorough methodologies for uncertainty quantification for neural interfaces. And I thank you very much for attending this workshop.
Thank you,
Do we
Esra
have
and
time for one
Antonino.
or two questions?
Yeah, definitely. We have a few already submitted. If anybody would like to add their question to the queue, please just pop it into the question and answer section. I'll start it off with a question about we'll start basic and then we'll go deep. So, can in silico studies help address the new or recent NIH requirements for complementary assessments to animal research? So can we use modeling and the OSPARC platform in the grant proposals and research applications to build the bridge between humans and other animal species?
So this has been done, yes. For example, some of the work Antonino has done has helped build model of porcine experiments to then predict on the basis of those validated porcine experiments translational expectations when moving to human data sets, and that was later on confirmed. In that sense, this has, for example, allowed some big companies to accelerate the process of moving from animal experiments to human trials. Other examples include the investigation how design of neural interface are related to resulting safety predictors, quite exactly like what we have shown today has been used by other large company in the recent past to accelerate the development and compress what would otherwise have been months of work into a few weeks to identify suitable designs. I just see also some questions on the chat. I'll try to address those. Thanks very much for asking. Yes, compound action potentials can be predicted on the basis of this kind of modeling. So the reciprocity theorem allows to leverage the same kind of simulations to relate neural activity modeling and exposure modeling to resulting fibers and to resulting signals. And we have a paper that is currently under review that shows how this can be done extensively and used also to optimize the signal information content of compound action potential. The question about extrusion in 3D that I see on the chat, the 2.5D model is a simplification. We didn't have to go into the limitation slide, but indeed, with the new data being collected both by the Feinstein team and also by the second REVA consortium, allows to build full 3D models that also accounts for tortuosity, for example, and for fascicle branching and merging, which turns out to have important impact as well on prediction. So among the uncertainty that is not covered here is the aspect of this fascicle merging and branching. And the last question that I also see in the chat concerns the question of the morphological and fiber data that needs to be collected. Most important is to have an idea about the statistics of the fiber diameters of myelinated and unmyelinated fiber. And if in addition there is topological organization across the cross-section of the nerve, such that some fascicles maybe have larger myelinated fibers and others few of them, that's helpful. That is something that of course can be obtained from histological data, but when you're trying to do that for a specific subject, that might be hard because you cannot section the nerve without losing your ability of properly providing therapeutic support. Instead, there are methodologies such as electrical impedance tomography that can be used to infer distributions of fibers in different regions. Those are, however, still relatively academic and haven't been shown to be suitable in clinical contexts.
Esra, we have another question that just popped up. Wondering how coupling between the FEM solver and the biophysical model are handled, given that the mesh and segments of the two models may not necessarily align?
Yes. So I'm not quite sure I understand that question. I'll try to answer it the way I understand it. So if this is about differences between the electromagnetic simulation and the real nerve, some of it is what I said just a moment ago about the electrical impedance tomography being a way of bringing in a closed loop manner, a way of adapting to model to what is observed as activity. So you can actually start with an imperfect model, observe signals from the nerve, and use that to improve the modeling inaccuracies. If it is about the alignment between the electromagnetic and the electrophysiological model, that's not really a problem in the kind of models that we have. We can use either in 2.5D anyway the cross section, and in complex 3D models that capture this branching and merging of fascicles, what we usually do is that we perform diffusion simulation along the different fascicles and then extract along the streamlines of the diffusion trajectories that can then be functionalized with electrophysiological responses. And in that way, it can be assured that the trajectory of neural fibers stays within the fascicle. I am not sure that I got the question correctly, this last one, and please feel free to contact me if I missed your real intention here.
Spectacular. I just want to close with one last question, if that's okay. I am floored that you all did a live demo today, and it worked so seamlessly and it was so quick. The surrogate model really is just a tremendous mechanism for anticipating the full effort that a full model would generate. But how do you test whether or not the surrogate model is a good approximation for that full model? I love the prototyping. I love that simulation restriction of that space. But what do you do after you feel pretty happy with that surrogate model? How do you know it's good?
One way of doing that is to do something similar to what people do typically in the fields of AI. And indeed, this has also aspects of AI in it, which is that you split your data besides the classic split into validation and fitting data. Here, what you can do is you can also take the data, put it into different bins, leave bins away, build a surrogate model out of the remaining data, and compare your surrogate model prediction for the bin that you left away to the full simulations that you have. And if you do that, for example, by splitting it into 5 bins and you do it for each of those, you get a pretty accurate statistical assessment of the error of your model. And depending on your surrogate modeling technique, for example, if you use Gaussian process interpolation or such techniques, they also provide an estimate about the uncertainty of the interpolation. So some of the surrogate modeling techniques are designed themselves to assess their own uncertainty. However, as is known, at least since the financial crisis in 2008, that was partly due to that kind of Gaussian process modeling. It's then critical to have an idea about how smoothly and how correlated parameter changes affect predictions. And if you don't have that, the uncertainty associated with the Gaussian process interpolation is also not very reliable. But the bootstrapping and cross-validation method that I first described is a solid way of assessing the quality of the model. And indeed, the tools that Javier showed provide such assessments out of the box.
Right. And that tutorial is available on the Spark portal, and as well as in OSpark as well. And people can register and request their own account to get to work on their own efforts on OSpark just by sending an email to support@ospark.io. So
and sparc.science, you will also find a detailed application example that can be followed that shows both the optimization of device design and the assessment of uncertainty associated with tissue properties.
spectacular. Never thought you'd bring it back to work in a relationship to the financial crisis, but that's why I love you, Esra. It's amazing. I hope that everybody had a really good time at the seminar, and that I would like to thank our panelists, Esra, Antonino, and Javier, for this delightful dive into in silico modeling, and I look forward to seeing you guys all tomorrow.
Thank you.
Bye. All the best.
Bye.
Bye. Thank you.